So what we're going to do here in this tutorial is we're going to learn about something called chi-squared. And chi-squared is going to be a new and our last important comparative statistic that'll tell us something about frequency-related data. So let's just get into it. I'm going to do that through testing for something called Mendelian genetics in a cross, just like we're doing in our fast plants. So let's just get to what we're going to talk about here. So what we have done already, or at least this worksheet says, just like our lab, is that there's been a true breeding cross between two homozygotes. As I show you in the paragraph above, capital Y is going to be a symbol we use for the color of the seed, and the T is going to be the allele that we use for the stem length. So we're looking at color, okay, and stem length, and I have that up here posted for your viewing displeasure. So we're looking at stem length and seed color, and as you can see, party people, we're going to see that the color, okay, yellow is going to be dominant to the green color. The green is going to be that recessive allele. It has to be homozygous recessive to show the green, and that'll be lowercase y. And a capital T will be for the tall dominant allele. Lowercase t will be for the dwarf, much shorter stem length of the plant. So what we have here is a true breeding cross that occurred already, okay, by the parental generation. And then philo 1, that's the offspring of this true breeding cross. What we have is dihybrids, and you can see they're both heterozygotes, meaning they all have the same phenotypes, just like our fast plants. Although we're not testing the same phenotypes and we don't have pea plants, we're doing essentially the same thing. So what we have here is we have yellow seeds, and what else? Tall phenotypes being expressed all the way through, because yellow and tall are the dominant, and both of these are dihybrids or heterozygotes. Okay, now we're going to cross these and we're going to see what we get and what we observe in our test when we cross them. And the thing is, we're going to get what we expect from Mendelian genetics, and we're also going to get what we observe, and we want to see how close these values come together. And that's where the chi-squared statistic comes into play. But it comes into play again when we have frequency. We're going to see how many of the different types of offspring, okay, are actually were made into seeds that could germinate. So let's get down to the little piece here. So we're going to do a dihybrid cross, and I want you now to fill in this Punnett square. Okay, and so I have YT here, but there are 3 other possibilities for the gametes, let's say in the male, okay, part of the flower. And then of course there is the same possibilities for the female. Okay, and if you got that, then I would like you to stop this video now and complete this table. So stop the video. Okay, so now that you have started me back up, you can hopefully see that you've got the same phenotypes as I do. Okay, so all we did was a simple Punnett square, and these are going to give us, very important, our expected. It's from the Punnett square that we get the expected. Now, we have to pull out all of the phenotypes. So let's go pull out what is yellow and tall. To be yellow, you have to be at least 1 dominant gene and 1 dominant Y and 1 dominant T. So to be yellow, you have to have a dominant Y, and to be a tall plant, not a dwarf, 1 dominant. So you can see that this falls in that category. So does this, so does that, so does that. Okay, and then certainly this has a heterozygous there, so that's still dominant. This, no, that's going to be a dwarf leaf. That's a recessive allele. This will work. Okay, this works. This works. These two, no, no, no. We have to have. Oh, there we go. And we're left there. So if you count 1, 2, 3, 4, 5, 6, 7, 8, 9, you can see where I got the expected ratios here. So yellow tall, you would expect to have 9. So that's where I get the 9 from. Okay. Yellow dwarf. Okay. Well, yellow dwarf, let's see if we got yellow dwarf here. And I'm just going to pick a different color for yellow dwarf. So yellow would mean I have 1Y and heterozygous. There it is. So yellow dwarf is right there. And or heterohomozygous, there we go, is another one. And the final one, I believe, is yellow dwarf is here. And that's where I would get also 3. And I put these in for viewing displeasure, and that's 3 for green dwarf. That'll be homozygous, so green tall. So there's my one green tall. There's my other green tall. And so these 3 right here are all green tall. Green because it got the recessive allele, homozygous recessive, and at least one dominant tall allele, okay, in that genotype. And when you're left with is one, what? You're left with the one that is green and dwarf. That should be an F there. And that's where you get the 1. And of course, that is going to be the homozygous recessive, and there's only 1 there. So you can see that my expected ratios, we've been talking about 9, 3 to 3 to 1 for weeks, and that's where we get it with a dihybrid cross, and we've seen that before. Okay, so how do I come up with the number of plants that are expected? Because I just found the expected ratio. Okay, so how do I do the number of plants that are expected? Okay, so what I do is, just to show this over here, is to do the following. Okay, and what I do is, and again, there's many ways to do this. So what I do here is take the total number of ratio values, 9 plus 3 plus 3 plus 1 is 16, a total of 16 there. So there's 16 parts, okay? And what we're going to say is we have 100 plants from the F2 generation. Don't lose sight of what we're doing. The F2 generation here is right here we're testing between the dihybrid cross. That's the F2. And so we're going to we raised 100 plants. What should we expect if the Punnett square holds true, which is tied to Mendelian genetics? So to find the actual number of plants that we expect from the Punnett squared, what I'm going to do is take this 16 and take the 100, divide it by 16 and find how many parts I have. When I do that, I get 6.25. Okay, and of course 1 times 6.25 gives me 6.25. 3 times 6.25 gives me the 18, and then that should give you the same number, right? 18.75. And then 9 times 6.25 gives me what? It gives me 56.25 when I do that in my calculator. So the number of plants expected from the Punnett square, which is basically following Mendelian genetics, that these alleles are on 2 different chromosomes, and we have independent assortment of these alleles and law of segregation. Okay, so if we have all of that working for us, we'd expect, okay, these group of genotypes to give us the ratio of phenotypes, and that's important. Okay, so now as we go on to the second page, I'm going to show you what we actually got. So when they tested 100 seeds from the F2 generation, which is basically the offspring of the dihybrid cross that we just did the Punnett square for, we actually got this. We got a 52 to 24 to 17 to 7. That looks a lot like, okay, the ratio that we'd expect, 56, 18, 18, 7. But is it exactly the same? Well, no, it's not. So the question is, how close am I? It looks like that I'm pretty close, but is it the same? It's not the same. So do we say that we didn't support Mendelian genetics, or did we? And this is where we need the chi-squared. And inherent in chi-squared is something called the null hypothesis. All right, so what we're going to do is we're going to test to see statistically if these frequency values are close enough to suggest that there is no difference. All right, we get observed 52, but we got expected, and let's put the E here for expected, 56.25. We also expect 18.75. Now you can't have 0.75 of a plant, but that's why the numbers broke out. All right. And of course, we'd expect 6.25. So how do we do this? Well, one way we could do is we could basically come up with a chart, and we could, let's say, measure the 1 to, let's say, make this 3 to 3, and this would be 9. Let's pretend it's to scale ratio. And then we could do it maybe a bar graph right next to it. So this is the actual 1 to 3 to 3 to 9, and then we can do the frequency maybe of what we actually got. Okay, we got something a little less. Okay, in this case, a little under. This one was definitely under, and this one was definitely more. Okay, and we could do that, but there's a problem, and we could do error bars to measure that, but there's a problem. If I do error bars for this value and compare it to this value, the error bars only tell me information how one variable is statistically significant to another. These frequency data are inherently tied to each other. If I make this number smaller and this number smaller and this number smaller, this number has to be bigger. Okay, or vice versa, many different combinations. These ratios are tied together. When we do error bars compared to some value or control, they're independent of each other, and that's not a good statistic. So standard error of the mean will not help us here with frequency data. We need something to measure if in fact these values of frequency are in fact close enough to these frequency, and they're tied together, as I just tried to explain. If you notice, we had the observed was a little more than expected. Okay, so so that's why this number is bigger, and then these numbers, 17 was a little under. Okay, and then this one was bigger, and this one was bigger. So the two of them, I guess I should have drew that red one to be a little bit bigger. But the point is, if these values were lower, these values would have to be bigger. They're tied together, so we can't do independent assessments of these values with standard error of the mean, the error bars. We're going to have to find another statistic, and that statistic is called chi-squared. Now, chi-squared is a variance, and I'm going to jump from this. We're going to go back to this shortly, but I want to go to my presentation. So hold on to your ponies. Let's go to the presentation. So here we go with a chi-squared test. Now let's not forget why we use it. We use it again to help with frequency data. Now we have frequency data on the worksheet we're using. I'm going to take a little diversion here to another analogous scenario. So here's another kind of a simplistic type of way to look at it. We have two sides of a coin, heads and tails. Those are the two types of choices or frequency data. Either we're going to have heads or tails, just like in our other data we have yellow tall, yellow dwarf, green tall, green dwarf, 4 choices. Here we have 2. And again, we can't compare the frequency relationships to each other using error bars because they're inherently tied to each other, as we just said. So what we have is this formula given to us in our reference table. So it says chi-squared. Don't worry about the squared, but you have this sum, looks like an E. So what we're going to do is we have this very simple test where we're going to flip a coin 50 times. Now we're going to expect when we flip a coin 50 times, because there's only 2 choices, 25% or 50% I should say, of our landing of the coin will give us heads, and 50% will give us tails. So out of 50 tosses, we'd expect half of them to be heads and half of them to be tails. Okay, now as you know, when you flip a coin or do anything of chance, it never comes out perfectly, especially when you have a small sample size. Now you know this to be true because you may know people in your family or people that are neighbors or other people that observe that you could have 3 children in a family that are all boys, even though it's a 50% chance to be a boy or a girl. Okay, so we also can see families that have all boys. Okay, so you can have all girls or all boys. I've known many families that happens that way. Even though there's a 50% chance to be boy or girl, how come we get observed that are different? Well, there are chances that you can roll or flip a coin or roll a die, okay, and get snake eyes 3 times in a row. You guys done that when you played games. There's a chance. But as you have a larger sample size, the expected values will equal themselves out. In terms of males and females, in the world, there's about 50% male and female. There's a little more females than males, only because males kind of wind up killing themselves in war more, okay, and other types of atrocities. And women tend to live a little longer. So that's why that statistic's a little bit skewed. But the point is, you'd expect and do get 50%. Now, in terms of flipping a coin, if you flip it 50 times, you may not get what you expect. But if you flip it 10,000 times, you can expect that you are going to get closer. Now, so what's a chi-squared? Well, we take a frequency type data like we have here, and we write down the observed that we get in the experiment. We actually flipped it 50 times, and 21 were heads and 29 were tails. We expect 25 and 25 because of the 50% chance. And then what we do here is we're going to take the observed and minus the expected. Okay, so 21 minus 25, -4, square it. And by the way, this should remind yourself of standard deviation calculation, because this is really a variance, how close and how far apart our expected is from our observed. In any case, we square it to get rid of the negative sign, and then we divide by the expected, which is 25. Now we're going to do that again for the tails. We have 29 minus 25, 4 again, but it's positive. Square it. If there was a negative, we take that away. And then we're going to divide that by the expected 25. Now, it's not always going to be even amounts. In our chi-squared calculation we're going to do in the worksheet, okay, it's going to be a 9 to 3 to 3 to 1. So it's going to be that 56.25, 18.75. We'll get to that. So this won't always be half and half. Just in this case, it is. Okay, so the clear thing here is, notice it's the sum of all of the what? Of the squares of the observed minus the expected values over the e. So we did that separately for each line, and then we add them together, and our chi-squared value is 1.28. Very important. Now, what does this mean? Well, it's sort of a variance. Now, again, it's helping us figure out something called probability, and I need to really try to make some sense of this for you. So please focus here. Chi-squared is a number that represents how well our values are really close together. We're never going to have observed and expected to be exactly correct. It's never going to happen. Why? Because when we do a lab, we can never have huge numbers that are just like the regular population. We're going to have some fraction of that. So by chance, we're going to roll snake eyes. We're going to have 3 daughters instead of having a family of 4. We're going to have 4 daughters and not have 2 boys and 2 girls. Because of those chance opportunities, our observed are going to be off. The question is there something beyond chance driving these numbers different than the expected? Is this coin tilted in a way that's a little bit heavier on the heads? Is this a trick coin? Or is these differences just due to the chance? That's what we're testing. So what we do is we take this chi-squared and we try to evaluate something called probability. Now, probability of what? Now, please listen. There's 2 things that can happen. We can have the probability that these 2 values are essentially the same, and there's just chance differences, meaning the numbers differ due to chance events, especially with small number of tosses. So we can have something called a high probability that these guys are the same. We could have a what? If it's a high probability that these guys are the same, then we can have a low probability that they're different, right? I can have this argument either one way or the other. Now let's just pretend for a second that these numbers are so close. This chi-squared is a small number. The smaller this chi-squared, the closer these 2 numbers. Really, isn't it about O minus E, the expected subtracted from the observed? As these numbers get closer together, essentially my calculated chi-squared, this should be a squared here, should be a smaller number. As this number gets smaller, okay, it's telling me that these guys are close, and there's a huge probability there is no difference between these numbers. Or there's a small probability that there is what? Very little difference. How do you want to think about it? I'll say it again. If this number is relatively small and these numbers are close together for reasons I explained, then we would say there's a high probability there is no difference. Or there's what? There's a small probability that they are different, if I didn't say that correctly last time. So which way do you want to work with? Do you want to work with whether they are different or not different? If I say, what's the probability that they're different? If they're different, we would say that there is a small probability that they're different, right? And we would say there's a large probability that they're the same. How do you want to phrase this? You can go either way because we're going to come up with probability. That's what's different here. We're coming up with probability from chi-squared. I'm going to show you. So to keep this from getting confused which way we're going to discuss this, we come up with something called the null hypothesis. And the null hypothesis is a statement that says essentially there is no difference between the numbers I observe and expected. Always. The null means nothing. There's no difference. See, we're making that statement. There is no difference, and therefore the chi-squared value is going to test that. So if we're going to say, hey, there's no difference with a null hypothesis, then, my party people, we're going to say that, hey, if this chi-squared is really small, okay, really small because these numbers are really close, then we're going to say that there's a high probability that there is no difference. See, I am fixing our discussion in terms of probability matching the support that there is no difference. The no difference is the null hypothesis. So let's make a null hypothesis for our worksheet. Let's go back there. And booyah, we're back here at the null hypothesis. So we're back to this study. So what can I say? Well, I could say maybe I can say there is no significant difference between the expected phenotypes and the observed phenotypes. And that's what the null means of the plants. I could phrase this either way, but the null hypothesis, party people, null means what? Nothing. Why are we doing this? Why are we making a nothing statement? So that we can test that statement to get something called a probability that tests that statement, that nothing. What is the chance that there is no difference between the observed and the expected? So we observe these plants, and here's our expected from our Punnett square. Okay, we got to come up with a probability value. And if that probability value we call p-value winds up being large, and I'm going to show you how to do that. If it's a large value, then we say that there's a great chance that there is no difference. See, the p-value is for the support of nothing. Why do we write a nothing hypothesis? Because it makes our discussion for the p-value fixed. We're always talking about the probability that nothing happens. Isn't there a probability that something's happening? Sure, but that gets confusing. So to fix our discussion, always the p-value, the probability value, okay, the chance, because nothing is definite, the chance that this value of chi-squared will give me a p-value large enough to say, hey, there's a great chance that there is no difference, and that, my friends, gives me too much a support for the null hypothesis, and we accept it, and we say that these guys are essentially the same. And what does that mean? That means that the only difference between them is due to the chance events that occur with small number of what? Possibilities, or in this case, flip of the coins, or in this case, movement of chromosomes from independent assortment. Okay, so the p-value gives me support for the null, so that I phrase and understand this, that it's the probability that nothing is different, okay, to keep it straight. So how do we come up with this p-value? Let's go back to my presentation. So we have in this example, a chi-squared. Again, a squared should be, for the flipping of the coin, to be 1.28. So how do we get probability? Okay, so what we do is we get our value. Again, I'm forgetting this squared part, 1.28. We have to think about something called a critical values table to actually attain a probability. Now, in order to use a table, we have to determine the degrees of freedom, just like we did with standard deviation. Because we have a heads or a tails, degrees of freedom are the number of categories minus 1. So because we have heads or tails, there's 2 categories or 2 frequency datas, we subtract 1 and have 1 degrees of freedom. Okay, so now that we have that, let's go to something called the critical values table. So once we have the chi-squared value, now we're going to go into the table. Now let me explain how we use this. Okay, now very simply, all right, what we do is we find our chi-squared, which is 1.28. Now 1.28 falls where? Let's go see where it falls in my table. All right, now it falls right there. Now let me explain. It's very important you understand. When the chi-squared gets bigger, hey, the difference between expected and observed is greater. If there is a larger chi-squared and these numbers between the expected and observed are large enough, shouldn't that lower the support that there is no difference in the null hypothesis? That should make sense to you. Okay, so think about this. This represents really 99.5% probability. It's huge. This is almost 100% certain. Can't be 100%, but close. That if, with 1 degree of freedom, if I'm in this area, if there's a 0 for my chi-squared, the smallest number possible, even the smallest number possible doesn't give me 100% because nothing is definite, but certainly if my chi-squared was so low that it was 0, we can say almost with certainty that there is no difference. All right, so if I'm 98%, okay, it's 0. Now, let's say my number starts to grow. Now I'm 90% certain. What's happening for my support of the null that there is no difference between numbers if the chi-squared number gets bigger? It drops, party people. It's dropping. I'll say it again. This is 99% or almost 100% support for the idea that there is no difference between the null and the expected. And that probability starts dropping when the chi-squared gets bigger. As the chi-squared gets bigger, the probability drops. Why? Because there's less of a chance that there is no difference. Hey, that's important. So as chi-squared gets bigger, the probability that there is no difference drops. So where do we lay a line in the sand and say, hey, there's too small of a support for the what? Well, we learned that 95% and 5% is a favorable line that people love to draw. So we get 1.28. It falls between 0.5 and 0.1. Well, that means 50% and 10%. We're somewhere in between 50% support and 10%. Well, that's way above the 5%. See, anything less than 5%, your number is so big, and your chi-squared is so far apart, or your expected is so far apart from the observed, that there's so little support for no difference that there must be a difference or a good chance of a difference. So the line in the sand is 5%. Subtract that from 100, 95 percentile. So anything above 5% in the 95% confidence range, hey, that sounds familiar, we're going to accept the null. So any probability that's above 5%, we accept the null as saying that difference between the expected and observed is close enough to say that there is no difference. And the only difference we're actually getting is due to chance events, okay, because of a small sample size, possibly. Now, if we were to get something like 3.9 that's lower than 5% or 5% or lower, then the probability there is no difference, we're going to say, is now too low, and we would reject the null hypothesis and actually state that there is an alternative hypothesis. And that means there must be something other than chance making this different. And that could be what you think that you're trying to drive the reaction. So the alternative hypothesis is what may be driving this. No difference? There must be a difference. Nothing's 100%, it's all probabilities. Okay, so very important that you understand that. So in this case, our chi-squared is so small, pushes us out into an area that's bigger than 5%, and that's big enough to say that there's support within a 95 percentile. So anything bigger than the 0.05% p-value means that we accept the null, meaning that we agree that there is no difference. That's the line in the sand. Okay, nothing is exact, but the larger the p-value, the more support for the chi-squared, and the smaller the p-value, the less support. So that's the line in the sand. Okay, so let's move on, and let's look at the chi-squared table that's in your reference table, because chi-squared, of course, is given as a formula. So we get 1.28. Our degrees of freedom are of 1, and notice the number as the chi-squared gets bigger. Hey, the p-value is small. This is a 5% p-value. This is a 1%. Anything that's in 5% or less is too small of a probability to support the null hypothesis. Everything is situated for the probability that there is no difference. We set it that way to keep it straight. Okay, so 1.28 is not bigger than 3.84, so our value is going to be in the larger range than 5. So in this case, we accept the null, that there is no difference. Okay, so, and what does that mean? You cannot reject the null hypothesis. The results have likely occurred simply by chance because the p-value is greater than 5%. We're in that 95 percentile. Okay, if you go back to your ant article, they also gave p-values. They didn't use chi-squared, but they came up with p-values, but it's based upon the null. So our tested hypothesis, and this isn't the hypothesis we write for a lab, it's a hypothesis we write when we do a chi-squared. You notice, if I reject the null because there is a large enough difference, the p-value would be, say, too small, less than 5%, we'd reject this null and say there is a difference, and say there must be something other than chance, which means there's some other factor. Maybe there's a independent variable causing it to happen. So now what I'd like you to do is take this and now actually do the work, okay, and determine the chi-squared value. So what we're going to do here, party people, is that you guys are going to solve for your chi-squared, okay? Take that value with the appropriate degrees of freedom. Remember, we had heads and tails and we -1. Here you're starting with 4 categories -1. So with appropriate degrees of freedom, take your chi-squared value and find your approximate p-value. Is your chi-squared going to be bigger than 7.82? If it is, the p-value is too small to support the null hypothesis, that there is no difference, and we reject and say there must be an alternative hypothesis. Or is your chi-squared going to be bigger than these values, and that means your p-value is going to be larger than 5% within the 95th percentile. And do you accept the null, meaning there is no difference and Mendelian genetics is upheld and the only difference is due to chance? So finish this out, stop the video, complete this, and then you can restart the video and I'll show you what I have. Okay, so stop it now. Okay, so I have my null hypothesis. I'm going to test it with my chi-squared. And if you notice, this formula here is given to me in a reference table. And if you notice, it's nice to do this in a chart. Here are my four, phenotypes, my four categories. Remember, this is frequency data. Here are my observed that was given to us. Okay, just like we're going to observe the phenotypes in our dihybrid cross in class. And then we have our expected that we got mathematically through the Punnett square with 100 plants. And then we, again, just going through this, observed minus expected, square it, divide by the expected, come up with a value. Do that for every single one of them. Now, unlike heads and tails, we're going to have different, expected values based on the 9 to 3 to 3 ratio. In terms of the heads and tails, that was just 1 to 1 ratio. Okay, so that's make sure you get the difference there. So they won't always be the same. So we take all of our values and then we sum them. That's why it's the summation here. And we come up with this chi-squared. Now you may have different decimal points here, which leads you to a little bit different answer, but no worries, it'll come out the same. So we take our chi-squared, that's 2.04. It's a small number, but let's see, with 3 degrees of freedom, because we have four categories. Remember, there's 4 categories here. All right, minus 1, we get 3, and we got a 2.4, and 2.4 is way above 7.2. It has to be 7.82 or bigger for the chi-square to be big enough, so far away from each other, the expected and the observed, that gives me a low enough support, okay, for the null. Because we have a small chi-squared, the expected and observed are too close together. And this degrees of freedom chi-squared is called a table that we look at, critical values table, easy for me to say, because our value is bigger. Our value of chi-squared is smaller than 7.82. Okay, our p-value is going to be way above 5%. And anything above 5% is a large enough probability that the null is supported and there is no difference. If we were to get a chi-squared that was 7.82 or bigger, then that would mean that we have too small of a probability to support the null. That's the cutoff here. And you should understand, as the chi-squared gets bigger, the difference is larger between observed and expected, and you should have a lower probability that there is no difference. So the support for the null is upheld. We accept the null because my chi-squared is above 7.82. Our p-value is way above 5%. And therefore, we say that the difference between the expected and the observed are the same, and the only difference that we're getting is due to chance events. All right, very important. In this example, we accept the null, and this is what we're doing in our fast plant lab. Any case, hope that helped.